Hi everyone, welcome to today's Open Box Science Genomics and AI seminar. And today it's my pleasure to introduce Sebastian Lobentanzer. I hope I say your last name right.
All right.
And he has a really unique background. He's a pharmacist turned into research software engineer. And Sebastian is particularly interested in this entangling causal relationship in molecular biology. And he does that by being part of a lab of Julio César Rodriguez at the Heidelberg University Hospital in Germany. And he has been there since 2021. And since 2024, he's also affiliated with the Open Targets Group at the European Bioinformatics Institute, EBI. He has received funding from two EU projects, including a translational quantitative project on ovarian cancer and another one where he received funding from DFG and Open Targets. Sebastian has been doing a lot of cool work, really building the knowledge graph in biology and connecting that to OON. And so I would love to, hear his talk as well. And so I'll let him take it away from here. Thank you so much, Sebastian.
Yeah, thank you so much for having me. I'm very happy to be here and talk about the things that we do. Um, I don't need to say much about myself anymore because you covered most of the basics, and so we can get right into it. Um, I will talk about biomedical knowledge economy, as I like to call it, because it relates to the entire process of ingesting information and representing it and then using it downstream. But I would actually like to start with something that's a little closer to my original education, up to the PhD that I did in pharmacology, because there is a huge focus on causality, particularly in treating patients with drugs that should intervene on their particular pathologies. And so this was also the objective that I had when I kind of set out in the postdoc. And I'm sure you've come across this idiom that correlation does not equal causation. It's fairly well established, but what does it even mean? Although it's superficially quite simple, in modern systems biology and systems medicine, it often poses a great challenge, particularly because of the dimensionality that we work with. And for example, if you look at the impact of nature versus nurture on any given trait or disease, you often and quickly hit what we call the curse of dimensionality. And then you can't disentangle correlation from causation anymore. And this is even complicated more by the fact that most of our modern experiments look like this, where we have a multitude of factors that go into this particular analysis, be it clinical parameters of patients or the specific conditions in a cell culture experiment, or you name it. And looking at the progress that we have made very recently with the new generation of artificial intelligence, particularly the neural networks, it becomes only natural to look to AI to help us make sense of these complexities and maybe drive forward the insights into these complex phenomena and facilitate our work in the same way that a diffusion model has facilitated creating this figure for me in like 2 minutes, which would otherwise either have taken me the better part of a day to create something that looks like this, or it could alternatively have looked like this if I did it in 2 minutes. And obviously, going beyond artwork in systems biology and medicine is much more challenging than just creating a nice illustration. And about this and related things, we have written a perspective recently for molecular systems biology, where we look at the spectrum of models that we apply. And this is a figure from that paper. On the left side, you see a human-created visualization of the effects of the EGF pathway. It is very, very clear, purpose-directed. You see EGF receptor, intracellular signaling cascade, the effects that it has, and some drugs that you could use to interfere with it. And then, just for fun, we wanted to see whether we could get GPT-4 and DALL-E 3, the top-of-the-line models at the time, to do something similar for us. And I think nobody will be surprised in any way that this doesn't work. and you just get a bunch of very attractive looking nonsense as an output. And notably, this is not due to a lack of knowledge on GPT-4's part, because if you ask it about the EGF pathway, it probably knows all the components, how they are connected, what they are, and what you can do to interfere with them better than any human expert alive, I would wager. But what doesn't work is the translation of this knowledge into a purpose-directed illustration, such as the one that you see on the left. And machine learning people will say it's pretty obvious why that doesn't work, because the training objective of DALI just wasn't that, because it's a diffusion model that's supposed to replicate some visually pleasing illustration and not worry about the mechanism behind things. But what's not obvious is how we would get such a system, how we would get a system that can stand in for a human designing a purpose-directed illustration. So looking deeper into this topic resulted in this perspective that I cite here at the bottom. And we looked at the spectrum of models that we apply in systems biology, from the very low parameter space, for example, what we use in randomized clinical trials, where we have a relatively manageable set of parameters that we are looking at, and usually you will use something like a linear model or a mixed linear model to estimate some effects there. Then you move into the more high parameter space, away from this, what we call the bottom-up approach of systems biology. Some other people call it the one gene, one postdoc way of working, because it's studying one particular thing to a great extent and then extending out from this one thing, like P53 in cancer or BRCA. And then we go to the high parameter models, the ones that enter the hundreds of thousands or millions of parameters, and we do different things with them. We have digital twins, or maybe a model that predicts the effects of perturbations. And then you increase the parameter space even more, and you end up with what we now call foundation models, which have parameter spaces that are so unimaginably large that you can't really grasp them, and that are supposed to derive generalizable principles, patterns, or mechanisms just by looking at a large amount of data. And this then enters the realm of top-down systems biology, which is more inspired by traditional machine learning and computer science, where you have a huge ton of data and you try to create an algorithm that makes some sense of it, that detects patterns, that learns from the patterns in the data directly. And then we looked at the way that we use knowledge in these models. How do we get the knowledge into the models? And also, what does this do to the biases in the model and the model's capabilities of inferring on causality? And what was surprising to me is that if you increase the parameter space to larger and larger models, it becomes less and less intuitive how the model uses the knowledge to drive some insights. So, in other words, you could say that the bigger the models get, the better they get at cheating your metrics or your training objectives, and just going routes that you wouldn't have taken if you gave the same data to a human. Now, changing gears a little, this was an example of a very complex thing, a very brain teaser-like thing that we think about in scientific work, and there are many others like it in the work of any scientist. You need a bunch of background knowledge for performing analyses about your model system, about statistics, about causality, for example. And then when you get some results, you need another set of very delicate knowledge, for example, about biology, medicine, pharmacology. And again, you need to have a thorough understanding of math, statistics, and these things. But there is another thing that we have to do in science quite a lot, which is more of the menial and tedious type of work. For example, we need to discover relevant data, we need to get it somehow, and we often also need to clean it a lot, as machine learning engineers will probably tell you. And then for interpreting results, you basically have to do the same things again. You need to look into the background of the genes that came up in your analyses, or you need to look at what a particular process does to a cell. And depending on who you ask, this may even be the majority of the time that you spend on any given scientific project. And this is essentially what is our main motivation for creating these frameworks that I will now introduce to streamline this work, particularly focusing on the menial tasks. So the first one that I want to introduce is called BioCypher. It is a framework to organize and ground knowledge in prior knowledge. And in a somewhat grandiose way, we call it democratizing knowledge representation because we want to really maximize the accessibility of these methods to the broad community, not just a couple of big labs and companies that have the money to hire people or entire teams to do knowledge representation. And if we want to describe it very high level, we could say it's an open framework that tries to do some central aspects of knowledge management. For one, we try to reduce redundancies in the data curation in the scientific community by providing reusable components so that if it's implemented in the open source way, it can be reused by others. We provide containerization to increase the reproducibility of these management pipelines. We want to reduce the time spent after making errors in manual implementations in Jupyter Notebooks, for example, by having a framework that's end-to-end unit tested with continuous integration. And we also want to facilitate grounding and harmonizing this knowledge based on the symbolic abstractions that we get from existing ontologies in any given domain. And this is what the configuration that we tease on the right here represents, where you have a specific domain for a knowledge graph. In this case, you talk about genes and diseases and some genes which are perturbed in some diseases. It is a modular framework, and it has three different components of this modularity. The first one relates to the input side. So you have some resources where you get the data from, and these may be in any kind of different formats, in any kind of different technologies. And we create what we call adapters to ingest these resources and standardize them for the framework to then operate on. And the second component that is modular is ontologies. So if you are in a given domain, for example, you're interested in genes and diseases, you could use gene ontology, disease ontology. You could maybe use the cell ontology or one of the experimental factor ontologies, depending on what kinds of data you're talking of. And we bring these together using this configuration that I've shown, where each class represents something that's really existing in your knowledge graph and that maps it unambiguously to the given ontology that's in the backend. And then we have the third component, which is the output side, which could be either very simple. You could stay in memory and have a pandas data frame or a network X object, or you could go all the way to a managed database management system with multiple labeled property graphs, such as a Neo4j database, and have your data there. And you can have many of them at the same time for the same project. And then you can use that to maintain and analyze your specific data, as well as archive it and also share it, for example, in the context of a publication. And now, given that you believe me that we increase productivity in knowledge management with this framework, we encounter the second problem, which is that we most of the time still need to use this knowledge to drive some process, to drive some creativity, to drive some insight, to maybe develop the next blockbuster drug or whatever your objectives are. And due to this, and inspired by the recent rapid progress in the large language model space, we created a second framework to extend the first one, which we call Biochatter, and which revolves around this conversational loop that everybody has by now gotten pretty much used to, which is a human user talking to a language model, and the language model hopefully giving some productive response to the question of the user. But as people have also found out in the recent years already, there are several ways in which these LLMs behave erratically or in which you can maybe improve the productivity by the LLM. And one simple way is to engineer the prompts to give you better responses. And what is easier than if we already know what kind of bioinformatics methods we applied, what kinds of technologies or what kinds of experimental backgrounds we have, that we standardize this and give it to the model such that the user then gets a more adequate response. But there are also more elaborate ways of improving the responses by the LLMs. For example, there is this thing called retrieval augmented generation that gets a lot of attention right now, that is supposed to counter the propensity of the LLMs for confabulations. So make up stuff that sounds very convincing, but that's actually not grounded in fact. And we naturally use the native connectivity between Biochatter and the BioCypher knowledge graph framework to natively connect the two and then enable people to interact with their knowledge in native language. But we don't only support knowledge graphs, but we also support the retrieval of unstructured knowledge, for example, from a vector database of papers where you can do semantic search and then use the results for retrieval augmented generation as well. Another handy feature of LLMs is that the newer ones can also parameterize APIs, which means that they can use external software for you. So if you're not in the mood to learn the X API, then you can get the LLM to parameterize it for you based on a request in natural language. And we can also go to the multi-agent setups where we, for example, have a chain of models that control each other's output, or where we even integrate with other modality, for example, by including a vision model in the mix. And in the same way as we did with the BioCypher framework, we would like to maximize the broad accessibility for as many research as possible. So we provide a bunch of different interfaces. We have the Python API, obviously, but we also provide developer toolkits for creating either a more prototyping style framework that is in pure Python. It's based on Streamlit. We call it Biochatter Lite. And we also provide a more elaborate infrastructure, which is based on a fast API, REST server, and then a Next.js application, which we call Biochatter Next. And I'm happy to show you a bit about them in a demonstration following the talk, but you can also see many of these yourself. The links you can find in the documentation at biochatter.org, which takes you to either the generic examples or to some vignettes where we have some of the use cases that I will explain in a minute. And also all of those are available in open source code repositories, so you can see how to implement such a thing yourself. So talking of these use cases, we have now as the main objective, as the main audience for these frameworks, the people who sit at the interface between the very computational and the non-computational users. So to give an example, I am the lead developer of these frameworks, but I'm also the working package lead in Horizon 2020 EU project that is called Decider, which deals with ovarian cancer, and we have multiple measurements. So we get the whole genome sequencing, transcriptome, methylome, and obviously clinical data of these patients that are treated in Helsinki. And we created a demonstration use case for the recent resubmission of the paper based on this cohort, which is in the context of a cancer geneticist that serves on a molecular tumor board, and given the patient, their individual history, and all their genetic mutations in the cancer, for example, they should decide whether they are amenable to a specific treatment, specific arm of a clinical trial. And this usually involves going through tons of data, going through additional references, etc. And so it seemed like a good use case to create for giving an example. And you can also see this in the vignette and as a live demonstration. We can check that later. So the idea would be that you have someone that is a domain expert like me in this Decider consortium, but also knows about how to apply this framework and then apply the framework in a domain-specific way for the given project using the libraries that we provide. So you would then create one of these front ends for your geneticist connecting to the Biochatter Python library, which creates, first of all, this main conversational loop between the LLM and the user. But obviously you have other things that you want to connect. So for example, we have this knowledge graph of the Decider cohort, which collects patients' clinical information, all of their mutations that they got from whole genome sequencing, the copy number variants that we collected, annotations from external resources, such as the druggability of certain variants, the biological processes associated with the genes that may be affected by variants, etc. And you connect this to the running database with the database agent. Then you may have an external tool. In this case, we are interested in drug use. So we use OncoKB, which is a public resource on the effects of cancer variants and their druggability. And then you could also have um, a multi-agent system, which in the use case isn't implemented by default. We have some modules for that, but it's very expensive. So you can have multiple agents that have access to the same tools that kind of double check or improve the results based on reflection and iteration. And then the main job of these developers of the framework is to set up the individual system for the user specifically. So we would, for example, instruct all of these models with their dedicated prompts. So you tell the primary model, you are an assistant to a cancer geneticist in the context of a molecular tumor board in the decider ovarian cancer cohort. You are supposed to help them with this and that. You have access to this and that. Please respond in this way and that way. You do the same with your databases. So you set up your knowledge storage, you set up your external tools, all given the developer toolkit that we have in the framework. And then you go into the details and you decide which LLM do we want to connect? Do we want to use a publicly available open source LLM, and which deployment do we want to use for that? Or do we want to use OpenAI because it's more streamlined and economical and maybe also more effective at the moment? Then you set up the system prompts, like what is the individual use case? What does the database agent do? What is the content of the database? You create, obviously, the knowledge graph, and we, in this case, also have a vector database where we embedded 30 recent papers about ovarian cancer for some background research. And we also connect the external APIs. In this case, we connect to OncoKB to, if some mutation comes up in the course of the conversation, the geneticist would be able to retrieve information from the OncoKB service. Importantly, most of this is open source, with the exception of the proprietary LLMs that you could use, for example, from OpenAI or Anthropic. And this is one of the major points that we want to make, is that open source is really necessary to keep up with the rapid developments that we see in this space at the moment. Related to the developments, we also think that we should be benchmark first. So what we usually suggest generally, and also in the implementation of these individual use cases, is that we, for each of them, develop a dedicated battery of tests that we can execute across the existing PyTest setup that we have, that has a matrix of all the different models that are there, all the different kinds of prompts that we may want to use, maybe different knowledge sources, RAG fragments that we want to test. And then we can continuously execute it and get the combinatorial results of all of these different combinations of tasks, prompts, models, etc., and monitor it. So you can see this on the website as well, but here is an example on a high level. This is the median accuracy across all tasks for all of the models that we had tested at this given time. And this is a comparison also between the closed and the open source models. On the very right-hand side, you see the open source models. You can see that they're usually very large, as far as we know. For some of them, we don't really know how large they are, but GPT-4 is estimated to be more than a trillion parameters. And we see that they still lead a bit in performance. Interestingly, about GPT in particular, GPT 3.5 Turbo in the newest version is the best model and not GPT-4, which may have to do with the system prompts, but we weren't able to look into this in detail because it's closed source. And then looking at the open source models, we see that, for example, there doesn't appear to be a correlation necessarily between the size of the model and the performance, even within the same family. And we have some contenders that are pretty good, so on par with some of the closed source models, for example, OpenHermes, which is a fine-tuned mixtral variant, and Llama 3.1 Instruct. And for Llama 3.1, it doesn't appear to matter whether it's the 8 billion or the 70 billion variant, interestingly, in our cases. And there are tons of insights that you can see from this data set, and we barely had a chance to look into many of the details there. We also benchmark specifically the functions that we introduce in the interaction between the packages. So for example, if we want to connect to a knowledge graph in BioCypher from Biochatter, we have this stepwise semi-deterministic approach to creating a query, which improves accuracy. So to explain, you could instruct an LLM, you could go to ChatGPT, you could say, I have this knowledge graph, you paste the entire schema configuration that you see on the right into the prompt, and you give it instructions like generate me a query to extract all gene disease relationships. And what we do in Biochatter instead is we do multiple individual steps where we first select the entities that will be relevant to the question, then the properties, then the relationships, sorry, then the properties of both entities and relationships, and then finally generate the query. And just by doing this, you increase the accuracy across the board, so across the closed and open source models by a lot. And finally, I want to give some perspective or vision for what I think we can do with open source frameworks and including large language models in the research process. I call it integrated research environment, a bit inspired by the integrated development environments that we use in programming that just aggregate a bunch of functions that you usually need for programming and thus streamlines your work. And if you think of a scientific project, for example, you're starting a PhD or a new position, you first familiarize with the background, which means you go through a bunch of papers, you look at some data, you need to catch up with what your PI did before, etc. And it would be nice if you could do this in a conversational manner. So you have an assistant that retrieves the right information from you from a huge stack of papers, and you can have a back and forth between the assistant and yourself, trying to clarify your understanding of the problem. Then once you're done with that, you could go into experimental design. And here I think it can be very helpful that these LLMs can assume different personas. And since everybody isn't an expert in everything anymore, um, we can, for example, complement our own expertises. So if you're a machine learning researcher, maybe you want to talk to a clinician to design your experiment. If you're a pharmacologist, maybe you want to talk to a statistician persona to make sure that you have the adequate power for your experiment. Once you have decided, you go into application. If this involves software, the LLMs can obviously help with applying that. And then in the end of the experiment, you obviously most of the time have a huge heap of results, which means you have lists of genes or processes, you have some images maybe, and you need to interpret this and go back to the original state of the art. And then you could, for example, have a conversation about whether this is in line with what was previously known about the phenomenon, whether this even contradicts something that was reported before in the literature, or whether this contradicts something that you see in some data that you have about similar experiments. Then you want to structure and critique your dissemination. And obviously, we don't want GPT-4 to write our papers, but it is nice to have an editorial persona that tells you, look, this paragraph seems kind of out of place. I don't really understand it. Maybe try restructuring, something like that. And I'm very happy that we're also in collaborations with some people from publishing, um, which means we can also facilitate the more menial aspects of peer review and maybe have an overlay that checks for initial correctness, or that maybe helps reviewer number two not to trash your research paper in uppercase letters, but rather give some more constructive and objective feedback. And this is the outlook. So we have 2 of 3 frameworks that are planned for the representation and for the application. We have BioCypher and Biochatter. We want to strengthen the interaction between these and also the community. And we want to additionally create a package that looks at the first step of knowledge management, which is the extraction of knowledge historically from texts. But also now we could think about multimodal learning, for example, including images. And we want to connect for this reason more closely with machine learning in the frameworks, for example, to go towards multimodal models, not only in imaging, but also maybe in genetics, maybe in single cells, or this kind of cell whisperer, talk to your cells kind of approaches, and in general, increase the robustness of data management in machine learning to go away from this typical, I have this script or this Jupyter notebook where I just do some manipulations on my data, and then a couple months later, nobody can reproduce it anymore. That would be the goal. for this part. And with this, I need to thank all the people that made this work possible, all the collaborators and co-authors, sponsors of the work, you for your attention and your kind invitation. And now I'm also happy to demonstrate a bit practically what these things look like and answer any questions that may come up.
Sure, thanks, Sebastian. Maybe we'll go into that demo first, and then people can
Yep.
think of questions and we can do Q after. Thank you.
Okay, so let me make this a little smaller maybe. Oof. Right. So there is this decider genetics use case that I talked about. We link it in the vignettes. So if you want to, if it's going a bit fast, you can always find the vignette on the Biochatter documentation and look through this yourself. So we have this repository which creates, in this case, two solutions. One of them is the low code Python only prototyping thing, and the other is this more elaborate one. So let's go with the more elaborate one first. This is at decidernext.biochatter.org. You can also find this link in the vignette. And this is what we envision the geneticist would see. It's a technical demonstration, so this obviously not meant for use in actual clinical diagnosis. And it contains synthetic data for the public demo, but we envision that we would have this welcome page here, which instructs the user what they can and cannot expect from each individual assistant application. And then we have some how to use instructions. For example, we give some example questions that we could ask. And in this case, we have, as I have alluded to, included three different ways of interacting with external information. So for example, we would have the knowledge graph. And also here, I included some copyable questions here. So for example, if it is about deciding which patients should enter a clinical trial because they haven't received a particular drug that would be relevant for them, there is this class of drugs called PARP inhibitors, which work best in patients that have a deficiency in homologous recombination, which you can figure out by doing molecular profiling. And given that you are a geneticist that knows about this, but that doesn't know the structure of the knowledge graph, you could ask this assistant, and then the assistant would come up with an answer based on the query that it generated, where we have an annotation that a patient is HR deficient and hasn't received PARP inhibitors, and then it receives it retrieves the patients for which this is true and gives some background information on the patient. So in this case, letrozole maintained 78-year-old woman with a BMI of 23.5 in partial response. And you can have a conversation about this. So you can go back and forth between the responses and the knowledge graph, or maybe the literature database, or even the OncoKB API, if you're interested in a particular effect of a variant. And I encourage you to try this yourself. Demonstrating it has limited value in this case because it's also already live. And then there is this other thing that I would like to show, which is generated from the same sort of backdrop. It's the same backend, it's the same language model configuration. These are usually created by having a Docker Compose setup, which you can then also put on a VM and present to people as an application on the web or in the intranet if you want to do that. And in this case, it is a very minimized variant of the Biochatter Lite interface, only showing one tab, which is a tab that we use to interact with the knowledge graph, just in a one-shot manner, where you can in more detail see what happens. So you can send this question here if you're, for example, developing the app and troubleshooting some queries or troubleshooting the behavior of your users. You can then try to create a query that didn't work for a user, see what goes wrong, and then maybe try to account for that. So you can do the same thing here. Maybe let's select another query. And this then does the same thing in the background, but what it does here is it gives you a modifiable query. So for example, if you are of the opinion that you would like to see not just the selection here, but rather the information of the entire patient. You could just modify the query here, run it again, and then you get no results because obviously I made a mistake in the syntax. Um, so you would be able to modify this and then develop in a back and forth manner the applications that then should ultimately be in the user-bound app that is on this side. And I guess I should stop there because it's all online and you can try playing with the models. You can try on your own, and maybe I'll answer some questions.
Awesome. Thank you so much, Sebastian. That's very, very interesting. And as people can see on the chat, feel free to ask in the chat box or raise your hand. Okay, so Tron has a question already. Have you seen Biochatter being used in real-world research? And you alluded to producibility. Are we guaranteed reproducibility because it's always anchored by a query? And how will researchers cite a result if they receive that from Biochatter?
Yeah, hi Trang. All good questions. Um, Biochatter being used in real-world research, it's probably a bit early to say. So the the application that I'm showing because I am part of the Decider Consortium, and we have a molecular tumor board application that we are creating, including our two frameworks. So we are building these knowledge graphs actually about the patients, not with synthetic data. And we're also developing a very elaborate and much bigger molecular tumor board than just this language model integration, but this will obviously be a part of it. And probably in the other projects that I am immediately aware of or involved in, even that is the most real world science research contexts that we have. And we have recently only resubmitted the Biochatter paper, so I guess it's a bit early in the pipeline to look towards other examples. Um, about the reproducibility, um, I alluded to reproducibility of the knowledge management pipelines. So that means Dockerizing your knowledge graph creation. And then by this way, having a transparent representation and a reproducible, deterministic way of modifying the knowledge that goes into the system. As per the reproducibility of language models, I don't think we can guarantee this because they are probabilistic to an extent. And also it depends on which language model you use. So even the different variants of the OpenAI models, if you go from one to the next iteration, they will probably give you a slightly or a very different response. And this anchored by a query, I guess it refers to the RAG mechanism, and feel free to just correct me if I interpret it wrong. But this is ongoing research. Like, I think there are no guarantees that RAG always leads to true responses. It's also philosophically a bit of a problem. But in essence, and this ties into the last question, how would you cite a result you received from Biochatter? I think Biochatter is a tool in the same sense that web search is a tool, or that maybe even more of these modern semantic search engines or Elasticsearch is a tool. It will retrieve something for you and it will maybe summarize and interpret it, and you can maybe even have a conversation about it. But ultimately, we need to make sure that we include the provenance, which we do in BioCypher by attaching provenance to each individual entity. All nodes and all edges should have provenance in a BioCypher graph. And in the same way, if you retrieve something from a literature embedding, the model will tell you which paper it took the information from. And this you can see in for example, the Biochatter Next. If you use the RAG, so not the KG RAG, but the vector database RAG, then it will, in this little widget, it will return the individual papers that it took the fragments from. And I'm not sure you would cite it in the same way that you probably wouldn't cite PubMed for having found a paper on PubMed. But you should cite the original research that it used if you drew some inferences based on the research.
Cool. Thanks. And the next question, our very nice talking demo. I was wondering, how does BioCypher and Biochatter handle sensitive biomedical data? Thanks.
So, well, I would say you're probably more interested in a practical answer. So you can run this completely on premises. With BioCypher, it's no problem at all. You can run this pipeline wherever you want, and you don't need to make it available to anybody outside of your network. You can run it on an isolated machine, even. And for BioChatter, it's a bit more subtle because that depends on which LLMs you want to use. If you are in this very agile and high performance space that you would use proprietary vendors like OpenAI for, which are still top of the line performance wise and also economically, then it's a bit of a problem. There is the possibility to have, for example, an Asia deployment of OpenAI, which makes it a little better, but I'm not sure how much better because you get your own server that deploys the model, but it's still, you can't access the weights or anything. But if you go into open source and you are happy with a little lower performance or a bit more engineering on your side, you can again run it completely in isolation. And we have a little section in the Biochatter paper as well that refers to this. And it is possible with some engineering effort to have any sort of security that you want, from the very open and agile way of just connecting to a bunch of APIs on the internet until the everything is on one computer and doesn't leave it ever. So yeah.
Great. If they ask, how much biology do you cover in human genomics? And for example, given two genes, could you link them with a therapeutic thesis?
So one of the biggest projects that we are starting right now with the ecosystem is funded by Open Targets. For the next 3 years, we will work on this knowledge management ecosystem that I have shown in the end. So the extraction, representation, and usage part will all be implemented for the Open Targets platform. I'm putting the link in the chat. And the crucial part about Open Targets is that it is an open source association with big ties to industry that collect tons of data for providing insight into the potential viability of drug targets and lead compounds for certain diseases. And we are aiming to create a flexible representation system, including a downstream language model-based assistant that can help the user in, for example, driving therapeutic hypotheses from the information in open targets data. So we are collecting these questions at the moment. As I said, the project has just started, but this is one of the bigger projects that we are starting in the ecosystem right now, and has the dedicated aim of facilitating high-level insights from all of these granular data that we collect on genetics, phenotypes, toxicity screens, etc., in this ecosystem.
Great. And as the other thing, I'm gonna try to ask a couple more technical questions and maybe bigger questions later. So yeah, Sebastian, I found your work because I was hacking through similar things, and you're so much farther along and better than I do. And I wonder, embedding, how do you do the knowledge graph embedding? How do you do the text embedding if someone output a input publication? Because I found that make a big difference,
It makes a huge
if you will.
difference.
So
Yeah.
how do you find those? Because I guess you show the benchmarking of the LM side of things, but I'm just wondering embedding model side of things.
Yeah. So we haven't looked too much from the generic point of frameworking. We haven't looked too much at embeddings because, as you say, the variability is huge, and it depends a lot on your use case, what exactly you want your embedding model to do. So usually we default, like for a long time, we used Ada 2 for the embeddings because it's just a nice general real world trade-off between accuracy and a wide range of topics that you can embed. ADA2, for the ones that don't know, is the previous generation OpenAI embedding algorithm that's kind of on GPT 3.5 level. They have new embedding algorithms, but I actually haven't had time to look a lot into these. And you could even go further and maybe look at these biologically fine-tuned models, which often have much worse performance, but as a trade-off, then account much better to the biological domain. And there's really no one size fits all. So it depends on the granularity of the questions that you have, how how you want to, in a sense, stretch your embedding space to maybe have a more resolution in the biological area if your questions are very domain specific about biology. I think for most of the service-directed applications that we do, a generic model always performs best because you can't predict what the user will want. And then by having a very constrained embedding space, you probably prevent good performance in other areas if the questions go in this or that direction.
Right. If Base is asking a related question, how do you see aggregation of multimodal data, image genetics at the embedding level or representation functions?
I'm not sure I completely understand. So is this about integrating data with a contrastive model? So having a model that is contrastively trained to embed two modalities similarly if they refer to the same real world concept, something like this. Um, yes, it's very promising. We have a pilot project in this publication sphere, peer review, preprint evaluation with EMBO, where we look at the relevance of figure captions, which in scientific papers often are said to encapsulate the essence of the manuscript, and then adding to this the panels of the figure and trying to extract more information by the combination of the figure panels and the captions simultaneously. And the models that I've tried, particularly ones from OpenAI, work very well for this. So they rarely make a mistake in judging, for example, if the caption is correct for the figure or not. But we are only in the very early stages, so there will be lots of things done in this space. And I'm particularly interested in the other modalities, such as if you can get some genomics, transcriptomics, or single cell measurements in there. So contrastive learning on the more high dimensional data and not images. So
Okay,
I'm hopefully optimistic, I would say, in this regard.
that's great. And I want to ask, what are you optimistic about this more general question? I think for a researcher using AI, you've seen probably a range of views from, we shouldn't use it at all, everything should be human produced, to hopefully AI will automate everything and we'll just retire and do whatever we want to do.
Hmm.
The Biochatter solution is kind of in the middle now. It helps automate
Yes.
quite some tasks. It still has human intervention involved. You can go and change the code. So
Yes.
I guess a few questions along the line, it's a more open-ended question. What do you see the human specific role to be in future of research in this whole ecosystem? And what is your, the ideal, because I guess the future is also part of what we construct it to be. What do you see as the ideal situation? And, like, I guess you're already demonstrating also some clinical use cases. How does that may translate to the clinical realm
Yeah.
that you also have background in?
I think an easy answer for this is to default to the regulators in the same way that we have the discussion about autonomous driving, for example. AIs can't be legal entities. I think most people agree on this. And we can't have an AI make decisions in the clinical realm anytime soon if we don't use them for a long time and we are very confident that they make less mistakes than the humans do. And then we need like a society-wide approval of this. So this is beyond the scope of what I see my work entail. I'm mainly concerned with the technical aspects of it and maybe the ones that are constrained to scientific process. And in this regard, I can only repeat that I think we should tackle the menial tasks first. So we also write in the paper, we want to supplement the, what we still don't understand about the human brain, that we have ingenuity, that we have creativity, that we can synergize very quickly or context dependently, right? These like highly creative tasks. I think LLMs may be able to simulate, maybe. There are some papers coming out now that generate hypotheses using multi-agent systems and then try to compare it, but I think it's still pretty early to say. And there is so much room that we can gain just by automating away the stuff that just takes away time from the things that we really want to do creatively or maybe interpersonally, like having discussions, taking some time to really reflect on something, and do away with the more repetitive, tedious, poring over a problem that is not really worth poring over kinds of things. And this doesn't exclude, for example, administration. We have one example on the website that has a non-biological use case, which is a project management system. So you would have a bunch of tasks in a GitHub project, and you would have an assistant that kind of aligns the team for the week's work, or maybe that takes a bit of um, form filling away from admin assistants, these kinds of things, right? Try to remove the less taxing things and leave the thinking for now to the humans, I would say.
Yeah, yeah, that's a great answer, and I think that's a nice way to conclude this. I think Sebastian, this is so helpful, and thanks for both the talk and the tutorial. So thanks everyone for listening, and feel free to follow us. We'll post the video shortly if you want to review, and obviously the links to Sebastian's work. So thank you all.
Thanks a lot for inviting me. Feel free to contact me. We have lots of contact information on the documentation, and we are very open source. So if anything is appealing to you or you would like to apply it, don't hesitate to reach out.